Today I'm going to talk about Asian fish diversity, and the question I'm going to ask is, are we there yet? And I'm going to provide a molecular perspective on Asian fish diversity. So in other words, do we actually know everything about the fish diversity of South and Southeast Asia? So this is the question I'm going to address today, but before I start with my talk, just let me introduce myself a little bit more. Rajaf has already done that, but here are some slides. I studied at the University of Zurich and Zoological Museum in Zurich, where I did my master's and my PhD. But already for my PhD and master's, I spent time abroad. I was in Belgium at the Natural History Museum in Brussels and at the African Museum in Tervuren. And I also had some research experiences at the State University of Stony Brook in the United States. As a postdoc, I went to the Natural History Museum in Madrid, and then I had a permanent post at the Natural History Museum as a fish researcher for a couple of years before I actually moved back to Switzerland, and I'm now based at the Natural History Museum in Bern. I started my career on these fishes that you can see here. So already as a kid, I was fascinated with the Lake Tanganyika cichlid. So for my master's and my PhD, I studied the adaptive radiations of Lake Tanganyika cichlids. But I also was interested in other cichlids. So for instance, I was working on a small radiation of cichlids from Lake Natron that occur in small thermal springs, as you can see here in the foreground. So in these small patches of water, you would see an immense number of these cichlids from the genus Alcolapia. During my postdoc in Madrid, I got interested in gobioid fishes, and I'm still working on this very fascinating group now. But it was in Madrid where I actually found my interest for Asian freshwater fishes, and that's what I'm going to talk about today. So in general, I'm fascinated by fishes. For me, these are the coolest animals in the world, and of course, with over 53,000 fish species, fishes are the most diverse vertebrate group. They come in all kinds of sizes, shapes, colors, and of course also flavors. And what is interesting is that actually each year, roughly 350 to 450 new species of fishes are being described. Fishes show a huge disparity in species richness. There are some families, as these three families listed here, that are immensely species rich, and other fish families are rather species poor. This diversity of fishes is also highly unevenly distributed across the globe. There are regions in the world, such as Australasia, where we have very few freshwater fishes, and then other places like the neotropics, where we have a huge diversity of freshwater fishes. Now, although the landmass of South and Southeast Asia is rather small, we have a very high number of freshwater fishes. And what is also very interesting is actually that we have a lot of different freshwater fish families. So in Asia, we have 121 freshwater fish families, 18 of which are actually endemic. This compares to roughly 45 to 50 freshwater fish families in Africa and 55 freshwater fish families in South America. So there's clearly something going on in South and Southeast Asia in terms of freshwater fish diversity. This is actually something you immediately see when you go to any fish market in Asia, the huge diversity of fishes on offer. And of course, when you see diversity on the fish market, you will eventually see this diversity on your plate when you have your lunch or your dinner during field work. Now, you look at this graph here. This shows the cumulative number of species through time. So we start in 1750 and we go to the year 2000, and we see that the number of known freshwater fishes has increased ever since. We don't see a plateau towards the present, which means that there is still a lot of new species being described on a regular basis. So we're still far away from actually knowing the true number of freshwater fish species in this region. This is from Maurice Cottola's 2013 publication, where he lists 3,108 species in Southeast Asia, encompassing this area outlined here. So, if we look not only at fish, but at eukaryotes in general, then we can say that over 2 million eukaryotic species are listed in the Catalogue of Life Annual Checklist 2017. However, there are estimates that up to 10 million eukaryotic species globally exist. This was predicted by a study by Mora et al. In 2011, where they found a predicted number of 8.7 plus minus 1.3 million. So up to 10 million species of eukaryotic species we might have on the planet. So that means that in spite of 250 years of taxonomic classification, still only a fraction of species is known. So there's still a lot of species to be discovered and to be described. Now, the question we have to ask ourselves is actually, how well do we know those few species that are already described? Do we actually know them very well? This publication here from Myrie et al. From 2018 gives a little insight into this question. So they looked at lizard diversity on a global scale. They found that 927 lizard species are only known from their type locality. So that's roughly 14% of all lizard species that are only known from their type locality. Of these 927 species, 231 are only known from their holotype, only from one single individual. The authors also found that 736 of these lizards were last seen alive when the holotype or type series was collected. Only 191 were seen alive after they were described. And of these 927 lizard species, 756 were observed in the wild in the last 50 years, that meaning since 1968, but still a high proportion, 171, were last seen between 1830 and 1967. So this little example from the lizard world just illustrates that even the species that are described, we don't know very well, and some of them we might only have seen once and never since. We go now back to the fish world, and I would like to introduce you to the Bardidae, the family Bardidae, the chameleon fishes. The first Bardidae species were described in 1822, so we have 2 species, and till 1957, not many new species have been described. In fact, in 1957, we only had 5 species described. From 1957 to 2002, we have a big jump. We're jumping to 15 species. So what happened here? Now, in 2002, we have the publication by Sven Kulan and Ralph Britz, where they provide a revision of the family Bardidae. They describe a new genus and 10 new species. So suddenly, the diversity of Bardidae is much bigger than we have anticipated before. So we thought it would be nice to provide a phylogenetic framework for the Bardidae, and we published the first phylogeny of the group in 2004. So in 2004, 16 species of Bardidae were known to science, and we were able to include 12 species as well as 1 undescribed species. So of course we thought, work done, we can lay back, but things have progressed since. So in 2004, we have 16 species of Bardidae, but now in 2019, there are 32 species listed. So a lot of new species have been described in the last couple of years, as you can see by this huge upturn in the cumulative species number. This slide just illustrates a few of the new Bardidae species that have been described in the last couple of years. And 2012 was actually a very exciting year for Bardidae, because Dario Urops was described. This was the first Bardidae described from the Western Ghats. So suddenly the distribution range of this, our understanding of the distribution range of this group has changed tremendously. all the previous Bardida species that we knew, they occurred in Northeast India, Bangladesh, or Myanmar, or Thailand. So suddenly we have Bardida in the Western Ghats. So we thought it might be quite the time to revisit the Bardida phylogeny, and we started a new project, and this is preliminary results. So we are currently working on a second phylogeny of the Bardida. So whereas in 2004 we had 16 species described, as I said before, at that time we were able to include 12 species and 1 undescribed species, but in the study that we have now, we know that there are 32 species of Bardida described, and we were able to include 24 species, but we were also able to include 10 undescribed species in the study. So in this, from 2004 to 2021, we made a lot of progress in understanding the diversity and evolutionary history of the group. On this slide here, you can see now the distribution of these different Bardida species. So each color dot here is a type locality or the locality where an undescribed species has been found. And what is very interesting to see here is this disjunct distribution. So we have here the species occurring in Thailand, Myanmar, northeastern India, Bangladesh, versus this group of Bardida species from southern India, from the Western Ghats area. And we are currently trying to figure out how we can explain this disjunct distribution. The second example I would like to give you now are the Charnidae, the snakecats. Now, snakecats, they have a very difficult systematics. It's riddled with taxonomic confusions. There are roughly 111 available names for snakecat species, but currently only 56 valid species of snakecats are recognized. 53 in the genus Jana from Asia, and 3 species in the genus Parachana from Africa. So this huge discrepancy between available names and valid names is probably due to periods of oversplitting followed by periods of lumping or even overlumping in the family Charnidae. Snakecats, they show a wide variety of body size, ranging from 12 centimeters to up to 1.8 meter. Many snakecat species, they show ontogenetic color phases. So what is meant with that? We can see that here, this is an illustration based on the publication from Benziger et al. From 2010. And here they show different ontogenetic color phases in the species Jana diplogramma. So we are starting here from very small juveniles, and we're going here as more adult they become, the different colors we have. So it's possible that in the past, these different color stages have been described as different species. So the first snakecat species was described in 1758. And then you see we have a gradual increase. And in this area here, we have a plateau. Probably one could have assumed, oh, we are about there. We know everything about snakeheads. But then after the year 2000, we have a sharp upturn. And in the year 2020, we have 56 recognized species of snakeheads. In 2006, the first molecular phylogeny of snakeheads was published. When this publication came out, I was very frustrated because I also was working on the molecular phylogeny of the group. So I was very depressed. I had sleepless nights, and what shall I do? What shall I do? And I have to say that after 2006, more molecular phylogenies of snakeheads were published. Also, in 2004, a group from Canada, from the Barcoding Center in Canada, published a paper on DNA barcoding in snakeheads. So what they did is they generated 121 DNA barcodes of snakeheads and took another 129 DNA barcodes that were already deposited in GenBank. So in total, they had 250 DNA barcodes of Snakecats. When we looked at their paper, we saw several problems. So the first problem was this one here. So this was an unidentified snakehead species from Sumatra, being sister group to all the remaining Jana species from Asia. And this is something that was hard to believe for us. So we thought, what's going on with this? Second problem was also the position of their Parachana africana. According to our preliminary results, Parachana africana is supposed to be the sister group of all the remaining Parachana species. So also something weird is going on here. So we teamed up with several experts in the field, and we did our own DNA reference barcode library. We generated 341 DNA barcodes ourselves and took 431 barcodes from GenBank. So in total, we were working with 777 DNA barcodes of Snakecats. So first to problem one. So that the Jana SP from Sumatra on the previous, in the previous study, we did some blast searches of that sequence, and we actually found not a Jana sequence at all, but it's a DNA barcode from a Raspora, something completely different than a snakehead. So this is actually a mistake probably in the lab. They mixed up some samples and they produced a DNA barcode of a cyprinid, and so it was a barcode of a snakehead. The second problem, Parachana africana. So what was going on with this one? These are our data. These are our Parachana africana sample. And as we expected, they are sister group to the remaining Parachana. So these are the Parachana africana. In the previous DNA barcoding studies, it's actually a mixture of Parachana obscura and Parachana africana. So these might be individuals that are not properly ID'd. In the meantime, when we did this study, more DNA sequences were submitted to GenBank and we downloaded them again. So here we have a slightly enlarged data set. And this in red here shows again this Parachana africana, Parachana obscura clade. And what we think is that this is actually until now undiscovered or undescribed species of snakehead from Africa, occurring in the vicinity of Kinshasa in the Democratic Republic of Congo. So it's a species that occurs in the Congo River and seems to have a rather limited distribution as far as we know, as we now know so far. Then I would like to put your attention here. While we were looking for sequences of Parachana in GenBank, we also came across actually some sequences that were labeled as Protopterus annectens, and the Protopterus annectens is a lungfish. So these sequences were generated during a DNA barcoding study in Nigeria, and it seems that they just mistook a Parachana of africana for a lungfish. So our data are here. As I said before, we used 777 DNA barcodes of Snakecats, and closely related sequences, we lumped them together into so-called barcode index numbers, so these bins. So in the first study on the diversity of DNA barcodes in Snakecats, 61 bins were identified. In our studies, we could add an additional 29 bins to the list. So all these gray ones are bins that were actually newly discovered in our studies. So as you can see, adding some new species, adding some new barcodes helps to understand the diversity of the group tremendously. If you compare the white patches here with the gray ones. So sometimes very deep genetic lineages were not present in the first DNA barcoding study and were only added by our study. So the red dots here, they indicate bin numbers that contain GenBank misidentifications. You see that in detail on this slide here. So we had several snakehead species in GenBank that we think were based on misidentifications. So we have, for instance, here 5 Jana barca in GenBank that are actually Jana blairi, or we have 5 Jana stewarti in GenBank that we think are actually Jana gachua. So 71 out of the 434 snakeheads from GenBank that we used were actually misidentified. So that means that 16.4% of the specimens in snakehead specimens in GenBank were misidentified. And that's quite a high number of misidentification. Subsequently to our DNA barcoding study, where we were able to actually include a lot of undescribed species, several of those undescribed species we included are actually described. And this is just a couple of examples of new species description coming out after our DNA barcoding study. But also what we found in our DNA barcoding study that is in widely distributed species, such for instance, Jana marulius, we have very deep divergent lineages that are geographically very well defined. And some of those deep split lineages actually had some available names. And after our DNA barcoding study, actually several papers came out that revalidated some of these species, mostly within the Jana marulius group, but also in the Jana gachua group. These are 2 examples from Sri Lanka, where actually Hiranya Sudasinghe and co-authors revalidated 2 snakehead species from Sri Lanka, Jana kelartii and Jana ara down here. So eventually, we also published our phylogeny of snakeheads. This happened actually just last year, so in 2020, we published the most recent phylogeny of snakeheads. We were pretty happy with our taxon sampling, and that's why we decided, okay, it's time to publish. I would just like to highlight one major finding of this study. So we have here a monophyletic Jana gachua group, and a lot of those species within the Jana gachua group are actually narrow range endemics in the Eastern Himalaya biodiversity hotspot. So in fact, 40% of all the Jana species are found in this region here. And this is quite amazing if you consider the distribution of Jana in Asia, ranging from Pakistan up to Russia. So 40% of all the species just occurs in this tiny area down here. It's highly likely that in the next couple of years, further snakehead species are being discovered and being described from this area here. So just to go back. So basically, it seems that we now know pretty well the diversity of snakehead species. But a big surprise came actually again in 2019. In 2019, a new genus of subterranean snakeheads was described from Kerala, South India. The genus was named Enigma Jana. In the year 2019, the first species was described as Enigma Jana gollum, and then a few weeks or months later, a second species, Enigma Jana mahabali, was described. This was a big sensation. And further studies on the morphology and the molecular phylogenetics of this new genus Enigma Jana actually showed that Enigma Jana is not part of the Janidae, but actually is so different from the other snakeheads that it should be placed in its own family, Enigma Janidae. And this is a paper we published in 2020. So based on these examples from the snakeheads, you see that although we think we might know a lot about a certain fish group, chance surprises or chance discoveries can actually turn our understanding upside down. And the nice thing is that it stays interesting. All these new discoveries, they lead to new research and to new exciting findings. I would like to switch now from Northeast India and Myanmar to Southeast Asia. I have been doing some work in Southeast Asia. This is the area actually in Asia I know most. So we're going to go to Sundaland. Sundaland encompassing Peninsula Malaysia and then the islands of Sumatra, Java, and Borneo. Sundaland has a very complex geological history. It started up 30 million years ago like a peninsula of mainland Asia. It's quite a stable block here. And then when we go to 20 million years, we have sort of a fragmentation of this block. We have inundation of certain land areas. When we move to 10 million years, we have most of Sumatra, for instance, inundated, so underwater, and only some volcanoes are sticking out. And then like 5 million years ago, the area has more or less the shape it has now. Sundaland is also very interesting when during the Pleistocene. So this is the condition now. We have the island of Borneo, Java and Sumatra laying on the very shallow Sunda shelf. The water here is very deep, very shallow. So during the Pleistocene, we had these climatic fluctuations. When we had the ice ages in the northern hemisphere and the southern hemisphere in the pole region, a lot of ice was trapped. The result was that the global sea level was much lower than it was today. Sometimes the sea level fell down up to 120 meters. So as soon as the sea level goes down, this marine area is getting smaller and smaller, and the terrestrial areas are expanding. So when the sea level is down to 120 meters, actually the islands of Borneo, Java, and Sumatra is actually connected by land to the Asian mainland. So we can say that currently Sundaland is in a refugial stage, whereas during the height of the ice ages, they were more expanded, more land masses. And of course, this exposed land was also traversed by huge river systems connecting the different land masses in Sundaland. In Sundaland, there are approximately 1,000 species of freshwater fishes being recognized. One of the biggest group of freshwater fishes in Sundaland is actually the Rasporina. This group with a very complex taxonomy. It's very difficult to distinguish the different species. Some of them look really quite similar. So in this project here, we sought to do a DNA barcoding study on the Rasporina. This was actually a project, part of the PhD of Arnie Sholiha, under the supervision of Nicolas Hubert. Arnie now defended her PhD, and she is working at the Bandung Institute of Technology. So in this study, we were actually able to generate 1,097 DNA barcodes of Rasporines. So in whole Asia, we are dealing with 140 species in 11 genera. In Sundaland, we have 79 species of Rasporina, and for our study, we were able to include 61 of those species coming from 189 sampling locations all over Sundaland. So this is the result of our study, and I just would like to focus on one small group here. So this is the genus Trigonopoma. There are currently 2 species described from this small genus, Trigonopoma gracile and Trigonopoma pauciperforatum. What you can see is we see here 3 main groups within Trigonopoma. So this might be something completely new, only occurring in Kalimantan. And then within this already described species, we see very deep genetic divergences. So we have here a very deep split into this group here versus this group here. And here we have several deeply divergent lineages. So we applied some species delimitation methods. They're shown down here that give you indications that you might or might not have distinct species. What we show here is a consensus of all these methods shown here in black. So it's possible that these different squares here actually represent different species. So this is something we often see in DNA barcoding studies. People generating DNA sequence data, they do some species delimitation method and says, oh, we have so and so many species based on genetic data. Now the question is, of course, does it really work? Can we do it like this? There's a huge amount of literature on species delimitation methods based on DNA sequence data, as you can see here on this slide. There are some people that say it works. There are some people that say it doesn't work at all. I would say it is a very good starting point. So if you go quickly back, if you have something like this here, and you have very deep split lineages, it's possible that this might be a different species than this. But of course, this is not something your DNA sequences will tell you. It just gives you an indication, and it gives you an indication where you should work with taxonomy to actually look for characters and whether or not you find characters that actually distinguish these individuals from these individuals. So I would not advocate only using DNA sequence data to describe new species, but it could give you some indications where it is worthwhile looking into more detail, where more hidden diversity might be hidden. One of the habitats I worked in Southeast Asia were the peat swamp forests of Southeast Asia. So peat swamp forests are unique habitats. These are waterlogged forests. They are not nutrient poor, and they are characterized by highly acidic black water. So we are talking about a pH as low as 2.5. That's almost as sour as lemon juice. Peat swamp forests provide important ecosystem services. The peat, so the organic material where these forests grow, can be up to 20 meters deep. So what is happening when a tree is falling down, it just lands in a very moist environment. It doesn't decompose and stays there. And over time, new trees are growing on top of it. So we have an accumulation of organic material and the forest on top of it. So these peat swamp forests are globally important carbon sinks over long time scales. So here we see a drainage canal in the peat swamp in North Selangor peat swamp forest in Malaysia. And you see here this thick layer of organic material below the forest. Normally the water table would be much higher, but this one is drained out, so the water table has already decreased. So the Southeast Asian peat swamp forests actually represent modern analogues of past coal-forming environments, like we had in the Carboniferous 360 to 300 million years ago. So in fact, Indonesia is the fourth largest coal producer in the world, and the coal deposits in Kalimantan are nothing else than actually Miocene peat deposits from peat swamp forests. For a long time, people have thought that peat swamp forests are very uninteresting. So this is some quotes from a paper by Daniel Janssen from 1974. Daniel Janssen is a very famous tropical ecologist. So he wrote in 1974, "The peat swamp forests in Brunei and Sarawak are notorious for their lack of conspicuous animal life." And he further said, "A collecting trip in this forest for animals of any sort or size was generally a waste of time." Now things have changed. In the meantime, we know that at least for freshwater fishes, these statements are not true. So as I said before, Sundaland harbors approximately 1,000 freshwater fish species. 200 to 300 species are recorded from peat swamp forests, and actually over 100 species are only found in peat swamp forests. They show a high taxonomic diversity of stenotopic blackwater species. Most species have been described in the last 10, 20 years. And what we know also today is that peat swamp forests harbor many miniature taxa. I will come back to that later on. So I was able to collect in some of those peat swamp forests over the last couple of years, starting in 2009 in Peninsular Malaysia, 2010 in Sarawak, 2011 in Sumatra, Bangka Belitung, 2012 in Kalimantan, and 2019 in Brunei. I just give you some impression how it looks inside a peat swamp forest. These are really beautiful habitats, but rather difficult to work in there. So some impression, and of course in these little streams you find a lot of very interesting species. This is how we collected fishes in those swamps. And eventually you got some fish for your studies. So this slide is just giving you a small overview of the diversity of peat swamp forest fishes that you can find. So I want to go into some of those groups in more detail, and the first group I want to present are the genus Parasphonemus. There are currently 21 species described in this group, and the important thing about this group is also that more than 5 species are critically endangered. So probably this number is even higher. So several Parasphonemus species are highly endangered. On this slide here we see them alive. So most of those photos are taken from the webpage of the Parasphonemus project. This is a very important and very important project. They are organizing captive breeding programs for endangered Parasphonemus species, but they also bring together people that have this common interest in this genus, because this genus is also very popular for aquarium. So many people are breeding them at home. So this is the phylogeny of the genus Parasphonemus. They are also called licorice gouramis for some obvious reason, as you can see here. The same stripes we have in licorice than we have in the fishes. And I don't want to go into detail with this phylogeny, but if you just look at the different color dots, you see that most species, so each color is a different species, most species have a very narrow distribution range. Some species are only found in 1 or 2 peat swamp forests in a very small area. Only few species, like for instance this one here or this one here, has a wider distribution. In many of the recognized species, we actually see very deep genetic splits, like here in Parasphonemus parvulus from central and south Kalimantan. We see some very deep genetic splits. This species here that we have as Parasphonemus sp. Kuching from Kuching in Sarawak was just described last year by Tan and Grinang. Another group that is typical for peat swamp forests are the genus Luciopcephalus, the picats, and the genus Spherichthys, the chocolate gouramis. These 2 genera are sister groups, and we're going to have a look now at each species individually. So first of all, we have Luciopcephalus aura. Distribution is limited in South Kalimantan here, but also occurs in Sumatra, probably also in the south here, but unfortunately I was not able to collect in this area here. And then Luciopcephalus pulcher, we have 9 genetic lineages that are geographically structured. We see some genetic clusters here and here, for instance. So each genetic lineage has a very narrow distribution. Spherichthys violanti and Acrostoma, 2 species with very narrow distributions. Violanti here and Acrostoma only occurs in this area here. I think I only was able to collect these species in 2 localities. Then we have Spherichthys celantanensis. We can distinguish 3 genetic lineages within this group. Again, each genetic group is geographically well defined, here encompassing, for instance, the island of Belitung and southwestern Kalimantan here. We have Sperichthys osphronemoides, consisting of 7 genetic lineages, and again, each lineage is confined to very narrow ranges. The interesting thing is that we have one population here in Kalimantan that is genetically very distinct from all the other populations. However, I don't think there are enough morphological differences between these populations here from Kalimantan and those from Malaysia and Sumatra to describe it as a new species. So probably one of the most exciting finds in the peat swamp forests of Southeast Asia was this genus here, Petacypris. So the genus Petacypris was described in 2006 by Kotela, Briggs, Tan, and Witte. So the genus Petacypris is also called the dwarf minnows. So at the time of description, Petacypris was actually the smallest, it's a very small genus, and one of the species, Petacypris progenetica, is actually the smallest fish species, with a female at maturity of 7.9 millimeters. So this is pretty small. This is compared to a coin from Switzerland. So these are really tiny, tiny fishes. Currently, there are 3 species described: Petacypris carbunculus down here, Micromegatus from Sarawak, and Progenetica, the smallest fish species here from the Jambi area in Sumatra. The differences between the 3 species is mostly based on pigmentation patterns, so that the 3 species, they differ markedly in the arrangements of pigment cells and pigmentation patterns in general. When you're in the field, it's very difficult to actually recognize them as Petacypris, because sometimes you have small fish in a net and you think, this might be juveniles from other species. And only if you look at them closely enough and you put them on your finger, you can see that this is actually Petacypris, adult Petacypris. So during my field trips, I was able to collect several populations of Petacypris. These are some examples of how they look in the photo cuvette right after collection. But this is the molecular phylogeny of Petacypris. So what we see again here, each color represents what we think are species. So we have 3 described species here, here, and here, and all the other colors we think are actually distinct species. We are currently describing them morphologically. It turns out that the pigmentation patterns are a little bit more complex than previously thought, so we need some more work. But the main thing here is actually, again, each species has a very narrow distribution. Yeah? Some of the species are actually only known from one locality. And we also see that the distribution of these different Petacypris species makes sense in terms of geography. So we think that actually these different species occurred through vicariance. So the division of a wider distribution, and then we just split off these peripheral populations. So we think that vicariance has played a major role in the evolution of this group. So as I said before, Petacypris is a miniature fish. So miniaturization is the evolution of extremely small adult body size. Yeah? Miniature fishes are defined as being sexually mature at the size smaller than 20 millimeters. What is also interesting is that a lot of miniature fishes show a trend towards reduction and simplification of various structures and organs. So what does that mean? Petacypris has a simplified skeleton. So here we have a skeleton of an adult Petacypris and comparing it to the skeleton of an adult zebrafish. Here are the sizes, 10 millimeters and 32 millimeters. These are clear and stained specimens, so we digest the muscles and we stain the bones in red and the cartilage in blue. So we see here in the zebrafish, we only see red, meaning we have everything is ossified. But in Petacypris, we have a lot of cartilage and very few bones. So compared to most cypriniforms and other teleosts, Petacypris actually lacks 40 ossification and has reduced size and or complexity of another 21 skeletal structures. This was published by Ralph Britz and Kevin Conway in 2009. So if we now compare actually an adult Petacypris to a juvenile zebrafish, we see that these look pretty much alike. So basically what we can say is that Petacypris is an adult trapped in a juvenile body. On this slide, we have a list of bones, and we order them by the time when they develop. So this is the size. So this bone, the first bone here, develops at the size of like 3 millimeters in a zebrafish. Yeah? So we'll do that for all the bones in zebrafish. So you see some bones develop very early on, some bones develop very late. If we now mark on this same scheme the bones that are missing in Petacypris in green, we see that some bones are missing here and there, but importantly, most of the bones that we are missing in Petacypris are actually missing down here. So these are bones that develop very late in the zebrafish. So for most of the bones absent in Petacypris, a relatively simple scenario of developmental truncation or progenesis can be evoked. So within the zebrafish relatives, we actually have two forms of miniaturizations. We have the so-called proportionate dwarfs. These are identical copies of their normal-sized relative. They don't miss any bones. And then we have the progenetic dwarfs, or the developmentally truncated ones. To this group, we count the genus Petacypris and the genus Danionella from Myanmar and India, and also the genus Sundardanio that is found in peatstone forests as well as Petacypris. So these guys are the interesting ones because they also show morphological novelty. So what do we mean with novelty? So in Petacypris, for instance, we have highly modified pelvic girdle and pelvic fin rays and associated muscles in males that possibly function as a clasping organ during courtship. So these modifications we only find in males. Females look relatively normal. In the genus Danionella, we have in males an anterior shift of the genital pore and the anus compared to the female that has the position normal. And then another one we have in actually in Danionella dracula. In 2009, we described a new species of Danionella dracula, and this species has this fang-like teeth. So these teeth are really a morphological innovation. Now you would say, what's so special about teeth in a fish? Because I know a lot of fish with very big teeth, like these examples here. But Danionella, of course, is a cypriniform, and cypriniforms are not supposed to have teeth. So what's going on? Again, histological studies showed that these teeth are actually not real teeth, but are actually extensions of the jaw bones. So these are fake teeth that are only found in males of Danionella dracula. So this is another morphological innovation, something very special we only find in Danionella dracula and in no other fish. And in Sundardanio, we have a croaking sound producing apparatus in males again. This is due to modifications in the Weberian apparatus, the fifth rib, the pectoral girdle, and associated muscles. So when you have Sundardanio in a net and you take the net out of the water, you can hear a sound like which is done by the males that produce this sound. So this is something you actually can hear yourself. So progenetic dwarfs are often considered taxonomic enigmas. They are often difficult to place phylogenetically because of their very simplified skeleton. One prime example is the genus Schindleria. these genus were actually placed for a long time in its own order until Johnson and Brothers in 1993 actually showed that they are gobioid fishes. Sundasalangs was often considered a relative of salmons until Siebert in 1997 found out that it's actually related to clupeids. Well, in Petacypris, for Petacypris, we at least know it's a cyprinid, but where is in cyprinids it belongs, we didn't know. So we started a phylogenetic analysis, and we could show that Petacypris is closely related to the Danioninae, to the zebrafish relatives. We further could show that miniaturization in this group occurred recurrently. So the miniature taxa are shown here in red and the normal ones here in blacks. So you can see that the miniaturization in red has evolved recurrently. Subsequently, other people have looked at the phylogenetic position of Petacypris and have come up with different hypotheses. So there's the hypothesis that actually Petacypris is the sister group to all the remaining cypriniforms. There is the hypothesis that Petacypris is actually a sister group to all the cyprinids. That Petacypris is somewhere nested within cyprinids. And the hypothesis that we favor is that Petacypris is actually part of the zebrafish relatives. So in summary, we have all these different phylogenetic hypotheses for Petacypris. Of course, we still think that this is the true phylogenetic relationship. So we think Petacypris is a close relative of the zebrafish. So the zebrafish, as you know, is a model fish. It's used in developmental biology, oncology, toxicology, neurobiology, regenerative medicine, and also stem cell research. Also, the zebrafish reference genome is one of the best known vertebrate genomes that we have. So somehow we thought that given that Petacypris is very interesting in terms of its developmental truncation, so it's a progenetic organism, it shows these evolutionary novelties. It is adapted to some very extreme environments like the peat swamp forest in Southeast Asia, and its phylogenetic position is still under scrutiny. It would be very nice to actually sequence a whole genome of Petacypris. None of us could actually do that, but we got in touch with some researchers from Norway, and what we did is we sequenced the whole genome of Petacypris and found some very interesting stuff. So first of all, we found that the genome of Petacypris is extremely reduced. So the genome of zebrafish is 1.4 gigabytes, but the genomes of Petacypris, the two species we sequenced, is 0.4 gigabytes. We don't have any gene losses. We have a comparable number of genes between zebrafish and Petacypris, but we can say that the Petacypris genome is roughly 3.5 times smaller than the zebrafish genome. In fact, it's actually one of the smallest fish genomes that we know. So as I said before, this genome reduction is not due to general loss of protein coding genes, but it's due to actually a very short introns. So in zebrafish, we have very long introns, but in Petacypris, we have normally very short introns. We also have a very small proportion of repetitive DNA in Petacypris. So this is the repetitive DNA landscape of Petacypris compared to the zebrafish, which is very huge, and to the normal other cypriniform fishes. What we also found is that we have an extreme loss of Hox genes and other developmental genes. Now, Hox genes are regulatory genes responsible for the segmentation of the embryo along the head-tail axis. We found that actually that in Petacypris, at least 7 Hox genes are missing compared to zebrafish. So these are the Hox gene clusters here in gray, and in red are the ones that are missing in Petacypris. For those with the question marks, we're not entirely sure whether they are there or not. So we don't have very good coverage of the genome in those genomic areas. So what we can say is that a normal cypriniform might have 49 Hox genes, but in Petacypris we have Hox gene loss. We only have 42 Hox genes. So now what we have to ask ourselves is, of course, if the reduction in genome size and the loss of Hox genes, is this restricted to Petacypris, so lineage specific, or could it be something to do with that Petacypris progenetic? So could it be that we find it also in other developmentally truncated taxa, this genome size reduction and Hox gene loss? Or it could also be that it might be a general feature of miniature taxa, proportionate dwarfs and progenetic dwarfs. So far, we have genome sequences of non-miniatures that don't show developmental loss and they don't show genome miniaturization. Our sequences from Petacypris, from a progenetic miniature, shows developmental gene loss and genome miniaturization. But the big question, of course, is what is with all the other ones? So we have a knowledge gap here. Do other progenetic miniatures also show developmental gene loss and genome miniaturization? Yes or no? Do maybe proportionate dwarfs show developmental gene loss and genome miniaturization? We don't know. And of course, what we also don't know is whether the extreme habitat where Petacypris occurs, the highly acidic black water peat swamp forest habitat, does this habitat has something to do with the genome miniaturization and developmental gene loss. So to fill this knowledge gap, we applied for money from the Swiss Science Foundation and we got funding to do this project. So in November, our PhD student Hiranya Sudhasinghe from Sri Lanka just started on the project. These are the project partners, and we have several project collaborators. We are really looking forward to solve these exciting questions. Now, this is how a peat swamp forest is supposed to look like. But unfortunately, when you go in the field and you look for peat swamps, you often find this. So what is happening? You have pristine peat swamp forests. They are drained, so the water table goes down. When the water table is down, the forests are cleared and replaced very often by oil palm plantations for the production of palm oil, which is a very important commodity for the countries of Malaysia and Indonesia. And of course, what you also have is that this huge carbon store that you have underneath the peat swamps, that this is actually going down. So you often have these forest fires where this dried out peat layer is going up in smoke, releasing huge amounts of CO2 into the atmosphere. So these carbon sinks are actually turning into carbon sources. So are we there yet? That was my initial question. And I can definitely say no, we are still a long way to go. We still have to discover a lot of exciting new species in South and Southeast Asia. So there's a lot of work for you guys to do in the future. So what can we learn from these examples I gave you? So I think these are some points that you can take home. You don't need to be the first to publish, yeah, except when you are describing new species. But if you're working on a phylogeny of the group, you don't need to be the first. What is more important is that you have a comprehensive taxon sampling. So this is really comprehensive taxon sampling is key to all the studies you are doing. And third, collect, because we need to discover those unknowns of biodiversity, and there's still a lot of them out there. So we need to collect more to get to know what is out there. And it's also very important to collect these things before they disappear. You have a huge, very fast habitat destruction going on, and a lot of species, especially those narrow-range endemics, are disappearing. And it would be very sad if they disappear before they have been discovered. Also, use GenBank data, but be aware of potential problems. Get out of your comfort zone, reinvent your research. I just wanted to show you that with the example of the genome of Petacypris. I didn't have any idea about genomics, and now I get to know slowly, I get to know how to do genomics. So every now and then, try new methods. And lastly, collaborate, because with collaboration, you get complementary expertise. No one can do everything these days. So you have specialists for this, specialists for that. So go and look for collaborators that can help you with certain methods or tools of analysis that you're not comfortable with. So with a slight delay, I'm at the end of my talk, and I would really like to thank all these institutions that helped with organizing the permits and the field trips in Malaysia and Indonesia. These guys here gave the money to actually do the collecting, and all these people here have helped in one way or another during field work in Southeast Asia. And of course, I also would like to thank all my co-authors on the many publications on Southeast Asian freshwater fish. It's actually not all the authors, but just a quick selection of people that have been publishing with me on Southeast Asian freshwater fishes. So I would like to stop with this slide. If ever you have the chance to visit Europe, please come to Switzerland, please come to Bern. We just opened a magnificent new exhibit called the Chamber of Wonders. It's actually in this exhibit, we bring part of our scientific collection from the basement up to the public. So we have a glass cubus, and you can walk around this glass cubus and you can look at the collection. Along this wall here, we house a reference collection of Swiss freshwater fishes. We have over 10,000 specimens here. So if you are interested in the freshwater fishes of Switzerland or Europe, please come and see this exhibit.